Hello everyone, welcome to my lecture on the next part is called as Cytoskeleton Part 2. I am Bendre. I am the Assistant Professor working at RBNB College, Sri Rampur. Today we are going to study the next part of cytoskeleton. In the previous lecture, I have been told you about the cytoskeleton, what are the types of cytoskeleton. In that I have discussed the cytoskeleton element that called as microtubules. In this next part, I am going to tell you about the next type that is called as microfilaments. So along with the three types, that is microtubules, microfilaments and intermediate filaments, this microfilament is performing a very important function in making the cell stable, in maintaining the shape of the cell. Okay, so let's see in detail about the microfilament. As the name suggests, it is microfilament. Micro means it is very small in size with filamentous structure. So in this lecture, I will tell you some points quickly. In the definition, let's see about the microfilaments. So these microfilaments are very small, intervened around each other. Okay, there are twisted structure. Okay, it is a small intervened twisted structure which is giving a mesh like a net like appearance all over the cell. Okay, and also twisted filamentous structure that form the part of the cytoskeleton. So in the cytoskeleton, the whole cell is covered by a mesh like network, filamentous mesh like network, which gives some rigidity to the cell. Okay, so next point is the structure. As I've told you, a single structure, if you observe the single structure under the electron micrograph of this microfilament, it is observed double stranded, just like DNA. Okay, it is double stranded, it is helical. Okay, twisted around each other, two strands are twisted around each other, giving a filamentous structure. Okay, in the next part is the size. The size is the smallest size, that is the 7 nanometer. As I have told you before about the microtubule, the size of microtubule was 25 nanometers diameter. Here the size, the diameter of the the microfilament is 7 nanometer. Okay, so after the size, let's move on to the monomeric subunits. In microtubules, we have studied that there are tubulins. In the monomeric subunits, there were alpha and beta tubulin that was forming the microtubule structure. Here also the same. This filamentous structure contains some actin globules. Okay, that is called as G-actin. Okay, why G-actin? Because it is globular in structure. It is spherical or globular in structure, while other one monomeric subunit is the F-actin, that is filamentous in structure. Okay, so hope you understood this monomeric subunit. Next is the nucleotide bind to the filamentous structure. Unless and until these nucleotides are not bound, there will be no polymerization taking place. Okay, so this nucleotide binding is very important for the formation of filamentous structure. So which is that nucleotide? That is ATP and ADP, that is adenosine triphosphate. So adenosine, after attachment of adenosine to the binding site of G-actin, the polymerization begin, or the formation of structure of microfilaments begin. Okay, so next is the function. So in the function, it carries out different kind of functions. Like first thing is the cytoskeleton, in that the microfilaments which are forming the net like, the mesh like appearance around the cell, it gives the proper rigidity and shape to the cell. Okay, so that shape is maintained by the microfilaments. In the next, the most important function here of microfilament is occurring in muscle contraction. In the muscle contraction, that is a very different part, big part, but I will tell you in very short that in muscle contraction, whatever the muscle, due to the muscle movement, okay, in the muscular movement, the whole contraction and relaxation that is taking place in the muscle is carried out due to the microfilaments, due to the actin and myosin complex. Okay, in the next, after muscle contraction, the next function is cytokinesis. In cytokinesis, the cell during the formation of cell, there is a next step called as cell division. When the cell divides, there is a pinching off of two cells. Okay, if there are here two cells, when there is cell division, here there is a contraction in the middle of the cell, which is forming a ring that is called as contractile ring. So this pinching off, when the cell divides, this pinching off into two cells is taken place due to the microfilaments. So microfilaments are taking a very important part in the cytokinesis, that is division of the cell. Okay, now next function of microfilament is the RBC. Why? What is the function in RBC? Whatever the shape of RBC, the biconcave shape of RBC is due to the microfilament. In the whole RBC, if you see, all the RBCs are having the same shape, that is the biconcave shape, that is only due to the microfilamentous structure. Okay, in the next part we are going to see about the endocytosis and exocytosis. In the endocytosis, when the substances are taken into the cell, that is only because of the microfilaments, which allow the external molecules to come inside the cell. Okay, in the next, that is the exocytosis. Whatever the material is prepared in the cell is taken out. That process is called as exocytosis. So in exocytosis, to take the proteins outside the cell, the microfilaments helps to form a pinching off of the cell. Okay, so this process is done with the help of the microfilaments. Okay, so after looking at all the functions, here I am going to next move to the structure of microfilaments. In this microfilaments, we will see the structure in detail. In this, what I have told you, that microfilaments is formed due to the assembly of G-actins. Okay, so G-actin are the monomeric subunits. In the monomeric subunits, first the G-actin, where it is formed by the cell. When these G-actins are formed, these are formed in monomers. Okay, so these are nothing but the proteins, actins. Okay, so first the cell forms the monomeric subunits, then afterwards there is polymerization of these monomers. Okay, so one by one this filamentous structure is formed due to polymerization of monomers. So in this, a polarity is maintained like microtubules. Okay, so this microfilaments are having a polarity. That means in this, if you observe this structure neatly, there is a barbed end. This filament is showing arrow-like structure. Okay, in this way. Okay, where this is the barbed end and this is the pointed end. If you will observe here, this is the barbed end and this is the pointed end. At the barbed end, this is called as the plus end. Okay, and the pointed end is called as the minus end. In the plus end, what happens? There is the assembly. Okay, there is assembly of or association of this G-actin molecules. So this is called as plus end. All the assembly takes place at the barbed end. Okay, and at the pointed end, there is disassembly. There is dissociation of the G-actin molecules. Okay, so this is forming the filamentous structure. Okay, after giving rise to the polymerization of filamentous microfilament. Here in the next point I am going to show you about that. In the previous point in the function I have told you about the muscle contraction. Here I am giving the example of actin and myosin complex. Okay, in actin myosin complex, same, this is the microfilament. Okay, in this microfilament the myosin molecules are attached. This is the actin filament. Okay, to this actin filament a myosin protein is attached. What happens when this actomyosin complex is formed? When there is formation of this actin myosin complex, there is contraction of muscles. Why? If you see the structure of myosin, there is a head, this is called as head, and this is the tail region. The tail region is attached to the membrane. Okay, the myosin structure, the tail region is attached to the membrane and head is attached to the actin complex, actin filament. Due to this attachment there is phosphorylation, and so the contraction, the process of contraction and relaxation takes place in the muscle. Okay, so this is called as actomyosin complex, which helps in the contraction and relaxation in the muscles. Okay, so in this, to summarize this microfilament, I would like to tell you again that the microfilament are very small filamentous intervened helical structure having size of 7 nanometer diameter. Here the monomeric subunits used are the G-actin and the F-actin. G-actin because they are globular in shape, so, and the yellow colored dots over here are ATP. With the help of this nucleotide, that is adenosine triphosphate nucleotide, the polymerization takes place, where the dissociation takes place due to the hydrolysis, that is conversion of ATP to ADP. Okay, so this was the structure, filamentous structure, polymerized structure of microfilaments, which is helping a lot, having main function in muscle contraction. I have shown you the example how the actin myosin complex takes place for the muscle contraction and relaxation. So along with maintaining the shape, the rigidity, the muscle contraction, the shape of RBC, endocytosis and exocytosis, the microfilaments, if they are not working properly, if they are not carrying out this function properly, or if there is any kind of mutation in the microfilaments, so there are numerous kind of diseases which takes place. One of it is during the cancer. Okay, everybody is known with the cancer disease. In this there is disorganization of microfilamentous structure. If there is change in the normal cell to the abnormal cell, all the microfilaments are disorganized, due to which what happened, it is converted, the normal cell is converted into a abnormal cell. Okay, and there is deformity in the proper formation of the cell and the shape. Okay, also when a drug like Cytoskeleton B is taken, that time it inhibits the structure, the filamentous structure of microfilament. Okay, so that time whatever the assembly, when Cytoskeleton B drug is taken, that time whatever this is the assembly and disassembly is taking place, it won't be taking, and therefore there will be no formation of microfilament, and then due to that there will be a cell death or deformity in the cell, which will give rise to different kind of diseases in the body. So this was all about the microfilaments, and next we will move on to the topic called as intermediate filament. So now we will move on to the next type of microtubules, microfilaments. Now we are going to study about the intermediate filaments. So let's see, intermediate filaments form a very important part of the cytoskeleton. As I have said before, that microfilaments, microtubules give rigidity to the cell structure in giving a proper strength to the cell, in maintaining the proper structure, circular structure of the cell. But the intermediate filament are the filaments that give it strengthens, it makes flexible, it gives a tough structure to the cellular cell. Okay, so in this we are going to see a very important part, that is the intermediate filaments. How is this? Let us see first the structure. So these structures are formed like rope. Okay, so the intermediate filament form a rope-like structure. It gives a twist to the filaments. Okay, and so why it is called as intermediate filaments? Okay, why it is called as intermediate? Because if you look about, as we have studied about the microfilaments and the microtubules, in microfilaments it was the smallest filament of the cytoskeleton. So microfilament has 7 nanometer of diameter, while microtubules had 25 nanometer diameter. But the intermediate filaments are having 10 nanometer diameter. Okay, if this is the filament, so this is the 10 nanometer size. So this is the intermediate size between the 7 nanometer and 25 nanometer. The size is 10 nanometer, not too large, not too small, and so it is called as intermediate filament. Okay, so the next is the location. So if we talk about the location, the intermediate filaments are forming mesh-like network just like microfilaments, but these are more smaller in size. But these intermediate filaments are occurring all over the cytoplasm, holding the cell organelles at their proper position. Okay, if their cell organelle is floating all over the cytoplasm, that floating nature to keep the organelle at very specific position is done because of the intermediate filaments. Okay, so this occurs in cytoplasm as well as in the nucleus. Most important location is they occur in the nucleus, below the nuclear membrane, in the nuclear lamin. So that specific structure to the nucleus is given by the intermediate filaments under the nuclear membrane in the nuclear lamin. Okay. So next is the subunit form in the intermediate filaments. So the alpha helical core forms this subunit. A single alpha helical form is the monomer. Okay. Then after to form the helical structure, the two monomers come together and form a dimer, and then at last they form a structure called as tetramer, where the polymerization takes place and they together form a tube-like structure that is called as intermediate filament, giving a rope-like structure to the filament. Okay. Next is just here I have shown you that there is no requirement of any nucleotide for the activation of the polymerization. Okay. As we have seen earlier, in microtubules we required guanosine triphosphate, while in microfilaments there was requirement of adenosine nucleotides. But in this intermediate filaments we don't require any kind of nucleotide for the polymerization to take place. Okay. Next, let us move on to the types. Okay. Here the intermediate filaments are of 6 different types. They are classified into 6 different types depending upon the amino acid nature. So these different types are made up of different kind of amino acids which are given for specific function. Okay, so as we know that it is present in the cytoplasm, it is present in cytoplasm of various kind of cells, and according to the structure of amino acid, there are this intermediate filament is classified into different types. So let us see the first type. So this is the keratin. So keratin, it is present in the epithelial cells of the skin. If you see here in the skin, we have a first layer called epithelial cell or the epidermal cell. In that cells, in this cytoplasm, the intermediate filaments are present, which is called as the keratin. In this keratin, there are two types, that is acidic keratin and basic keratin, which forms the hard keratin and the soft keratin. The soft keratin are present in cytoplasm, while the hard keratin helps to form the structure. Okay, just previously I have told you that the keratin form a typical structure of the cell organelle. Okay, so to give a proper rigid structure, the intermediate filaments helps a lot. So this keratin are present in the epithelial cells in the cytoplasm. Okay, which form the structure like hairs, nails, which are the hard keratins. Okay, so this protects the epithelial cells, okay, to maintain the proper structure. Next, after the keratin, there is the desmin. Desmin intermediate filament are present in the muscle cells takes place for process called as contraction and relaxation. Okay, so along with the microfilaments, intermediate filaments also helps a lot in the contraction and relaxation process. Okay, with the help of desmin filaments. Next is the vimentin. Vimentin helps to strengthen the cell, to help the make the cell strong for any kind of disturbances that is taking place in the cell. So that structure is maintained with the help of vimentin. Also, it maintains the location of the organelles. Then, for giving a specific structure location in the organelle in the floating cytoplasm, the intermediate filaments forms the mesh like network in the cytoplasm, where endoplasmic reticulum, nucleus and mitochondria are held for to maintain the proper position in the cell. So that is due to the intermediate filament called as vimentin. Okay. Next is the neurofilaments. These neurofilaments are present in the nervous system, that is in the axons. The axon structure is maintained by the neurofilaments to transfer the signals from one position to other. Okay, so these signals are moved with the help of the neurofilaments, and the structure of axon is maintained by the neurofilaments. Okay, so next filament is the laminin. So laminin, as I've said you earlier, that the filaments present in the nucleus, in the nuclear lamina are the laminins. So these laminins maintain the structure of the nucleus, okay, and it protects the nucleus from any outside disturbances. Next is the nesting. Sixth and last part is the nesting, which is present in the nerve cells. Okay, here in the central nervous system, all the nerve cells are formed with the help of the nesting intermediate filaments. So these are the very important types. Okay, you should remember this type, because in different kind of net set exams or any other competitive exam, this importance of these types have been asked and their functions. So it is important to remember these functions of all the types. Now next we will move on to the assembly of the intermediate filament. How they are assembled. How is the formation of the intermediate filaments. Here I have told you earlier that it is forming the helical core. Okay, helical strands are produced, okay, of the intermediate filament. So from single filament there is polymerization of different filaments, and then there is formation of rope-like structure. Okay, so here first thing is the monomers are formed. Alpha helical monomers are formed, which are having N terminus N and a C terminus N. N terminus NH2, that is amino, and this is carboxylic N. So this single helical structure then forms the diamond. Okay, this two helical structure. Here you can see there is attachment to the opposite side. Here there is N terminal, but the other strand has opposite terminal at its end. Okay, so for the attachment of tight attachment of the filament, then these filaments form the alpha helical twisted structure. Then they form a diamond, and these diamonds the addition of second dimer forms the tetramer. This helps to polymerize the structure, and together they form a very strong, rigid, rope-like structure. Here you can see there is an anti-parallel arrangement. All the strands are not arranged start to end position in the same direction. First of all, polarity is maintained, means if there is an N terminal at start end, there is a C terminal also at the start end, and like this there is an anti-parallel arrangement of the filaments to make the structure like this. So in this structure, in this single structure, these tetramers are put together to give like this different tetramers form together and form a rope-like structure. So hope you have understood that from a single monomer, dimer, tetramer, a rope-like intermediate filament structure takes place. This intermediate filament help to make the structure of the cell very rigid, very tough and flexible to protect the organelles and maintain the shape of the cell. Okay, so hope you have understood all the three types of the cytoskeleton, that is microtubule, microfilament and intermediate filament. So thank you for watching.